Hi everybody, this is Dr. A, and this lesson is going to be an overview of the conventional cultivation and systems for identification of microbes. Alright, so cultivation, which is growing the microbes, it is required for identification and characterization of certain, at least bacteria and fungi, although now with more modern methods, we have gotten a lot better at direct testing on specimens, which we'll talk about that. And so, we have some direct or quick testing available, but it still doesn't cover everything that we can find and everything that can cause infections. So the purposes of cultivating or growing the bacteria or fungi on some, plates or on some media is to isolate the microorganism that is present in a clinical specimen so we can identify it so we can better treat the infection. Within the growth that will happen on the plate, we also need to be able to determine which of these is likely the culprit for the infection and which of these might be innocent bystanders such as part of the normal microbiota. You need to also be able to obtain sufficient growth of the clinically relevant microorganism, the one that's causing the infection or the problems, to then be able to go with further with the test of identifying and characterizing it, especially, for example, as far as antibiotic sensitivity, so that you know the right therapies to give to your patient. And when we cultivate, we need to meet the nutritional growth needs of the organisms. So you have fastidious and non-fastidious organisms. And if these terms, aren't familiar, think of it fastidious as like fasting, okay? So fasting as in not eating. So fastidious organisms are the ones that are picky eaters, and so if you give them the wrong media, they'd be like, no thank you, I'm not going to eat that, I'm not going to grow, right? So they would fast, they would not eat. And so they're really picky at what you give it. You have to give it just the right nutrients. Non-fastidious, they're like, yeah, just whatever you have, I'll grow on it, it's fine, I'll eat whatever. And so they're a lot easier to grow in on general media. There are several forms that media can take: liquid, solid, and semi-solid. The liquid is exactly that; it's liquid, so you can mix it, and it's a broth media. And when the media is uninoculated, it would be clear. And so then if there are bacteria growing in the media, it is turbid or cloudy. So you can, with automation, you can use turbidity measurement, turbidimetry, so looking for cloudiness, nephelometry, which looks for light scatter, or you can do pH indicators indicating that there's metabolism going on, and that can cause a color change that indicates bacteria. Solid media, which is probably what you think of when you think of media, is the plates, the agar plates. So the solidifying agent is agar, and it makes the bacteria will grow on top of, or the mold or fungi will grow on top of. And so it provides for the isolation of pure colonies if you do your Strict method correctly. And you can also use it to identify and look at the colony's morphological characteristics. So what does it look like growing on that particular agar? And of course, the agar is kind of like the base, and on that you can put the different nutrients, and you can cause different types of media agar to be made depending on what nutrients you mix with agar. Now this is not done by hand anymore. This is usually done by the manufacturer, and then they send you these plates already made. Semi-solid agar is a broth that contains a small amount of agar, so it helps create a diffusion gradient. It can restrict oxygen diffusion, resulting in the potential identification of the oxygen concentration that promote growth. I have a slide here that's going to show you this a little bit better. So when we're talking about oxygen concentrations that promote growth, we're kind of over here. So if in your broth or your semi-solid media, like a motility media, for example, here is a good semi-solid example of a semi-solid media. And so in the motility broth, well, it's not broth, it's medium, motility medium, you stab here the organism into it, and if it's non-motile, it will stay in the stab. If it is motile, it will go throughout the broth, but the broth is not so thick that it can't go through it, right? That's why it's semi-solid. But even in broth cultures, if you just pick it up without mixing it, you can see, and especially if it's in a tube like this, you can see where the growth is happening. So obligate aerobes will grow towards the top at the surface of the broth. Obligate anaerobes will grow at the bottom. Facultative anaerobes will be more concentrated at the top but will grow throughout the tube. Aerotolerant anaerobes will grow throughout the tube nice and evenly from the top to the bottom. And your microaerophiles will grow just a little bit below the surface in a concentrated area there. Okay, so this is broth liquid, and so you can see how it's clear if there's no growth, and it's turbid if there is growth. This is an example with concentration of antibiotics with the too low a concentration not inhibiting the growth of the bacteria, and therefore there's cloudiness and turbidity, and the higher concentration of the antibiotics inhibiting the growth, and therefore the broth here is clear. And then here's an example of agar plates in a solid media. So again, agar is kind of like the base, and in the liquid, liquid can be the broth. There are different things that we can mix into or make them into. So we can make them into enrichment media. So they contain specific nutrients or chemicals that can enhance the growth of particular pathogens. So your buffer charcoal yeast extract agar is an example there. We can use that one for, I believe, for Francisella tularensis. That's one example. Thioglycolate broth, although we use that as a good like holding media, extra media. Nutritive, they support the growth of most non-fastidious organisms. So tryptic soy agar is one of them. Like blood agar is tryptic soy agar with blood added to it. But a nutritive is just a regular, tryptic soy agar. You can buy just that, and most organisms that are non-picky eaters, non-fastidious, will grow on it. If it is selective, it inhibits all organisms except those that are selected. So it could select for only allowing gram-negatives to grow, or it can select with only allowing gram-positives to grow, for example. So it could contain compounds such as bile salts, that's what's in McConkey, dyes, and antibiotics. Antibiotics could inhibit some of the normal flora or different things, or specific antibiotics could inhibit specific types of bacteria from growing, right? So your PEA agar, phenylethyl alcohol agar, is an example of those, and this one is used for to inhibit gram-negatives and allow staph to grow. And then it can be differential, which allows colonies of one species to be differentiated from other bacteria that are growing on the plate. If it is, it's usually based on metabolism, right? So it's a visible characteristic using an indicator dye or chemical reaction. So it could be, for example, on the McConkey, if they ferment lactose, the colonies turn a very bright pink. Or, for example, on the mannitol salt agar, if it's the Staph aureus, the part of the agar where it's grown will turn yellow from being kind of pinkish color before, if it's a non-staph. McConkey agar is one as a combination, is both differential and selective. So it selects for gram-negative rods, and it allows for a differentiation between lactose fermenting and non-lactose fermenting. And then you have the environmental requirements to grow these bacteria or fungi. And so oxygen and carbon dioxide availability are the two things that we can control. Most clinically significant organisms are facultative anaerobes, meaning they can grow in an anaerobic environment, but they can also grow with oxygen. The temperature, most bacterial pathogens like body temperature, so they like 35 to 37 degrees Celsius. Some might require cold enrichment, and at 20 Celsius to even all the way to 40 Celsius. And the pH here, so most clinically relevant bacteria prefer a near neutral pH, 6 to 7.5. Some might be able to grow at extremes of pH. So moisture contents, humidified incubators are used to maintain adequate moisture levels so the agar doesn't dry out and they have enough humidity in the incubator. Next is isolation streak. So a common technique is the quadrant method, where you would inoculate your specimen in the first quadrants, as indicated here with the swab. So putting some of the specimen, could be a drop of your blood culture bottle or whatever it can be. So we inoculate some of the specimen. If you have multiple plates to set up, you need to inoculate your specimen on each plate first, right? And then spread that inoculum over the first quadrant. So on this plate, this is the initial inoculum, and if you use a metal loop, you would spread it out and then you would flame the loop to kill any bacteria on there, let it cool off, and then you would streak your second quadrant. Now, this picture, even though the position of the quadrant is correct, I don't like because there's not overlapping. Now, you're supposed to overlap into your first quadrant, right? So, how much overlap, it depends on how heavy your culture is, or how heavy, how many bacteria are in your initial inoculum. And sometimes it's difficult to assess, right? And so if you inoculate too heavily, you could end up with heavy growth all the way around, and without having good isolated colonies, you may have to do another round of plating to try to separate out your colonies. But either way, you rotate, so you flame your loop, rotate the plate, and then flame this and then let the loop cool off, and then streak the next quadrant. And then you would flame your loop again, and you rotate, and you streak the next one, and the next one, and then you can go. You can do it you can do it up to 5 times here. Most people say you just do to 4, so that's the bare minimum. The idea is you're thinning and thinning and thinning the streak so that at the end, in that last quadrant, you have isolated colonies. Now be careful when you streak the last quadrant to not touch these here, so be careful. I use the light and angle your plate so you can see the streaks that you're doing. If you're using disposable loops, then you should swap loops in between quadrants. Otherwise, again, you may have too heavy of growth on the plate. And the goal again is to have isolated colonies like here, where you can see the individual morphology of each colony. So how do we identify bacteria? So we have genotypic criteria. So this is some of the newer methods where the bacterial genome is characterized using molecular techniques. So for example, the BioFire does that. And the presence of a specific gene is used to identify the organism. So in genus and species, there's certain genus that have specific genes present, and then the other genes vary by the species, and so that's how you can get a definite ID. And those methods test for those genes that are unique, right? Not the ones that are all in common to all bacteria, for example. And then if you use phenotypic criteria, you are looking at observable physical characteristics like what it looks like when it grows on the plate, so the colonial microscopic morphology. And then if you make a gram stain off of that culture, that colony on that plate, then what does it look like, in the microscope? So you have a gram-positive, a gram-negative, you have cocci, you have rods, what do you have? And then you can also assess its metabolic characteristics. So that's the biochemical identification. And so part of it, an environment required for growth. Did it grow well at 37 in a regular incubator with regular oxygen? Did it need an anaerobic environment? Is it susceptible to specific antimicrobial agents? And we can test them against specific ones. What were its nutritional requirements and metabolic capabilities? What kind of sugars did it ferment? What kind of amino acids did it use? Did it produce a certain gas, or did it do this or that? And so the metabolic identification panels there are different panels that exist depending on if you're trying to look at for gram-positive bacteria or gram-negatives. And so you can set up these plates or these cards to get your definitive ID depending on its metabolic and nutritional capabilities. So again, for genotypic identification, so this is usually a direct detection within the specimen that is received. So if you wanted to do a respiratory panel on a biofire, you would use, the like sputum, like the pneumonia one, and it's direct detection of the nucleic acids without growing the microorganisms on a plate. So they're looking for either the presence of a single gene, multiple genes, or the partial sequence of a gene that makes that specific bacteria unique. It does not indicate that the organism was viable in that specimen, that you could have cultured it, but it does indicate that it was there in the specimen, right? And so you have to, the doctor has to interpret, of course, the results within the context of everything. One thing I do like about these genotypic identification panels is their capacity to test for bacteria and yeast and sometimes even parasites, all in viruses and all of that, all in one panel. The sequencing methods determine the base sequence of a gene or multiple genes or the partial sequence of a gene. And you can even do whole genome sequencing of the bacteria to get the whole genome. And so some of those where you want to do whole genome sequencing stuff might be more used in epidemiology, where you're tracking mutations and you're tracking specific infections and things like that to especially if they're linked to contaminated products. And then the primary culture methods is a phenotypic identification method, and that's really what micro's been doing for decades. The molecular stuff has only really been on the scene for, I would say, a few years, 10 years probably at the most, like really actively being used. And so it's a routine set of media that is used usually to isolate and identify pathogens. And so I'm sure each lab has its own like protocol set, but there are things that are pretty standard. And usually you have, wherever you set up your cultures, there's usually a cheat sheet that's posted and saying if you have a wound, you need to set up this, and this type of media, these plates, this broth or whatever. And if you have a respiratory specimen like a sputum, you set up these. And so you set up the plates based on the site of infection, which means based on the type of specimen you received. Is it a urine? Is it a wound? Is it a respiratory specimen? Is it a biopsy? Like, what is it? And this is all driven by the most likely infecting organisms present. And you should usually include some kind of nutritive media, some selective and differential, and enrichment or backup broths. And so a combination of all of these to catch, all the most likely organisms. The primary plate reading is the reviewing of the growth present following incubation of that routine set of media, often done for at least 12 to 24 hours before we start looking at them. And then you want to start correlating that microscopic colonial morphology with the microscopic morphology. So if you did a gram stain off of the primary specimen, what did you see in that gram stain? And then is that what's growing on the plate? And then if you gram stain that colony, is that correlating with what you saw on your direct or first gram stain. This indirect one would be the one off of the colony. And so that once it's all correlated, you're like, yes, this makes sense, then it helps you to determine the likelihood of the pathogen identity, and then it helps you also to know what biochemical identification to set up. So for colonial morphology, some of the things that we look at are the size. So is it pinpoint, which is from 0.5 millimeter to maybe 1 millimeter. If you're not familiar with metric, you probably need to go get familiar with metric and find a visual reference of what that looks like. Small is 1 millimeter to up to maybe 5 millimeters. Medium is about 5, and then large is 5 and up. So pigmentation is what color it is. Does it have pigment? Is it yellow? Is it golden? Is it white? Is it gray? What is its shape? So form, elevation, margin. So is it circular? Is it irregular? Is it convex? Is it flat? Western margin, is a margin entire or straight? Is it a rose? Is it lobate? And you have different ways you can describe these. The density or opacity, so can you see through it or not? Is it transparent, translucent, iridescent, or not? Or is it opaque? The texture, is it smooth or glistening? Does light reflect on it, and is it shiny, right? Is it rough or dull? Is it dry? Is it wrinkly? The colony consistency you get more when you touch the colony with a loop. So is it butterous, meaning is it buttery? And think buttery as in warm butter, right? A butter that's been left to sit at room temperature for a while and very nice and soft when you hit it. So a lot of colonies can be that way. Is it friable? Does it crumble like a crumbly cookie? Is it mucoid? So when you touch it, you pick up mucousy strands a little bit. And are there changes in the agar, especially if this is for blood agar? So for the hemolysis change are read on blood agar. Is it alpha, beta, or gamma hemolytic? We'll go over that. If there's pH indicators in the media, did it change colors? Is it pitting of the agar, etc.? So those are all things you want to assess when you're looking at your colony morphology on that plate. So here are some examples of some small colonies here that are a very white-ish, almost kind of gray, but it's very, very nice, clear, crisp white. They're glistening, they're circular with entire margins and stuff. I would say they look slightly convex, but sometimes you have to angle the plate to be able to see that. Group B strep here, we see some beta hemolysis, some clear areas around the colonies. These are small to pinpoint colonies. Here, this micrococcus has some small to medium-sized colonies, and it's a pretty yellow color. Brown colonies, entire margins, etc. So you get used to the terminology and how to describe all of this with practice. You can also, when you have the colonies that grow, instead of doing a biochemical ID, you can take the colony and use MALDI-TOF to identify it. So you would place part of the colony on this MALDI-TOF plate along with a matrix and other things on there to prep it properly. And so you can put one colony per little circle, and then you can load several of them on there, and then you have to identify in a computer where you've put the different specimens, and then you can run it into the MALDI-TOF analyzer right here. And so what happens in the MALDI-TOF is that the specimen will have a laser beam that hits it. It breaks the specimen, the bacteria in there, up into pieces that get ionized. So fragmented and ionized, and those pieces will enter the mass spectrometer to detect, and they'll start flying up towards the detector. The smaller charged pieces will fly faster and hit the detector faster, and the larger pieces will fly slower. And so this is a time of flight mass spectrometer, meaning it is measuring the time it takes to make it through a specific length of path to the detector to identify the organism. And as it's being detected, you'll get hits here like this. You get these peaks at different mass units and mass to charge ratios here. And it generates a mass spectrum, which is kind of like, to me, it kind of looks like a city skyline, right? And each organism has a specific one. It has a specific look, just like each city has a specific skyline look. You know, each organism has its own mass spectrum that is unique to it. And so then this is compared to a computerized database of all these mass spectrum for all of these organisms. And what's neat about this is this database can be updated as we find new things or we find variants and things like that. So you're not required to memorize these, although you probably will get familiar with the ones you see the most. The computer does the work and the matching for you. And then we also have some enzymatic tests that can be done. So some of these are rapid tests, easy to do. You can do them straight off the colonies that are growing on the plate. The catalase test can differentiate staph from strep. And you put hydrogen peroxide, you put some of the colony on slide, add hydrogen peroxide, and watch for bubbling. If it bubbles, it's catalase positive, and it's likely staph. And if it does not bubble, it is catalase negative, and it's likely strep. The oxidase test differentiates groups of gram-negative bacteria. The oxidase gives a purple color. This is oxidase positive, this is oxidase negative. And it identifies if a microorganism has a cytochrome C oxidase enzyme. This is used in the electron transport chain. Then we have the urease test. It can help identify some of the members of the Enterobacterialis family, so like E. coli and all of its friends. But for example, it can be used for H. pylori. So if you've ever seen the test for H. pylori that's done off the biopsies, so the physician, they'll do an endoscopic procedure and get a biopsy from the stomach ulcer, and they'll put it in the media that is a urease media. So initially the media looks like this with the biopsy in it. So it's on a card, right? It'll look this yellow color, and then if H. pylori is present in that tissue specimen, the organism will hydrolyze urea into ammonia, water, and carbon dioxide and turn the media where it's in a bright pink color, and that's considered positive. But this is not the only one that will do that, but that's how the urease test works. For indole, it presumptively identifies E. coli, and it does this red color change here for the indole test. Then we have the PYR test, which is pyrrolidonyl arylamidase. Easier to say PYR to me. It helps to identify strep pyogenes and enterococcus. You can do it straight off the colony, as here you add your reagent, PYR reagent, and if it's positive, it will turn color. You also have the hippurate hydrolysis. It helps to identify Gardnerella vaginalis and strep agalacticae, which is group B strep. So this will be from vaginal specimens. Campylobacter jejuni and Listeria monocytogenes. So Campi is a GI infection. Listeria can cause GI infections, but also meningitis and other things. And as hippuricase hydrolyzes hippurate, it produces glycine, which makes a pretty purple color. And so, again, the metabolic pathway assessment is a phenotypic way to identify bacteria, and it's based on visible changes, color changes, if they metabolize a certain thing. So if they oxidize some carbohydrate or ferment some carbohydrate, of course the oxidative process does require oxygen, the fermentative process does not. And so we use oxidative fermentative medium. A lot of that's built into these little metabolic panel that we can use. It can degrade certain amino acids. And so it determines the ability of a bacteria to produce an enzyme that either deaminates, dehydrolyzes, or decarboxylates an amino acid. And then single substrate uses, the organism can be inoculated on a medium like citrate, malonate, or acetate and observed for growth or detection of certain metabolic end products. Of course, I feel like metabolic pathways are just well assessed now in some of these tests. So this is a quick, like the API 20E for gram negatives. So this is what API strip would look like if it's completely negative. There's no growth, no reaction in any of the wells. And then here is what it looks like if all of these are positive. So this is your negative key, this is your positive key, basically. And depending on the patterns and the reactions in each of these wells, you can help identify different types of gram-negative bacteria. Here is another version. This is the microscan version, where this portion right here has different metabolic pathways that are assessed. Some of them are sugar, some of them are amino acids, etc. And then this area, a lot of this area here, right here, is antibiotic sensitivity. And then it's good to also talk about the rapid identification schemes. So these are also often called just algorithms. There's quite a few of them out there. But for example, for this one, if you look at, start with gram stain morphology. If they're gram positive and they're cocci, and if it's catalase positive, it's likely staph. If it's catalase negative, it's likely strep or enterococci. If it's gram positive, bacilli. If it has spores, it's bacillus. If it doesn't have spores and it's catalase positive, it's Corynebacterium or Listeria or others. If it's catalase negative, it's likely Lactobacillus or Actinomyces. For the gram negatives, if they grow on MacConkey, it's probably one of the Enterobacteriaceae or Pseudomonas. If it does not grow on MacConkey, it could be Haemophilus, Brucella, or Legionella. If it grows on MacConkey and it's oxidase positive, it's likely Pseudomonas. If it is negative, it's the Enterobacteriaceae. If it's a gram-negative cocci and it grows on Thayer-Martin, it's going to be a pathogenic Neisseria. If it doesn't, it's going to be another Neisseria species. So these are some. Here's another one just for gram-positive. So I like this one too because it gives a little bit more depth to the algorithm. So of course you start with catalase. You start with catalase test on a gram-positive cocci, and if it's in clusters, it's catalase positive. It's likely a staph. And if it's in chains or, small chains and stuff like that, it's likely a strep, and it'll be catalase negative. And then you can go down this road where if it's coagulase positive, it's staph, coagulase negative, but oxidase positive, it's Micrococcus. If it's oxidase negative, and then you can check with bacitracin here too, and novobiosin to determine do you have staph saprophyticus, staph epidermidis, or Staphylococcus lugudinensis. If it's catalase negative and bile esculin negative, and then you alpha hemolytic, we're looking at strep pneumo or viridin strep, depending on its octotion's sensitivity or resistance. If it's beta hemolytic on blood, and depending on its bacitracin sensitivity, it could be group B strep or strep pyogenes, which is group A strep. If it's bile esculin positive and it has either alpha, beta, or gamma hemolysis, but it lacks salt, it could be Enterococcus. If it doesn't lack salt, it would be non-Enterococcus group D strep. So this gives you one for that, and then we even have one for gram negatives. So for this one, if it's diplococci, that's aerobic, that does not utilize maltose, it's Neisseria gonorrhea or Moraxella. If it utilizes maltose, it's Neisseria meningitidis. If it's a cocobacilli, you're looking at Haemophilus influenza, Bordetella pertussis, Pastorella brucella, or Francisella tularensis. If it's a bacilla and it's lactose fermenting, yes. If it's slow fermenting, it's Citrobacter serratia. If it's fast, it's E. coli, Klebsiella, and Enterobacter. Then, of course, you have to do further tests to identify these guys. If it is non-lactose fermenting, but it is oxidase positive, it's Pseudomonas. If it is non-lactose fermenting and oxidase negative, and it produces hydrogen sulfide, then it's Salmonella and Proteus. If it doesn't, it could be Shigella or Yersinia. And then we have the comma-shaped ones that are oxidase positive, depending on the media it grows. We have Campylobacter, Vibrio, and H. pylori. So that wraps up this quick lesson on the identification of, conventional identification of microorganisms, and I hope that was helpful to you.